Through their study, researchers have successfully mapped the various systems within the brain that regulate emotions, offering potential targets for therapeutic interventions. Greetings everyone. Today we will be delving into an intriguing subject in our video. A recent investigation conducted by Dartmouth researchers has unveiled the existence of various systems in the human brain that are responsible for regulating emotions. This groundbreaking study has provided valuable insights for potential therapeutic interventions. By differentiating between the processes of emotion generation and emotion regulation, the researchers were able to pinpoint specific regions within the anterior prefrontal cortex and other higher-level cortical hierarchies that play a crucial role in regulating emotions. These findings represent a significant advancement in our understanding of emotion regulation, as previous research had not been able to isolate these areas with such precision. Moreover, the study shed light on the neurochemicals that interact with these emotion regulation systems, notably receptors for cannabinoids, opioids, and serotonin, particularly 5-HT2A, were found to be abundant in the regions involved in emotion regulation. So, without further delay, let us dive into our discussion. Have you ever had the urge to let out a scream on a truly awful day, but somehow managed to hold it in? This ability to suppress our emotions is thanks to the incredible power of the human brain in regulating our emotional responses. It plays a crucial role in helping us navigate the ups and downs of everyday life. As we observe the events unfolding around us, our capacity to adapt and reframe the situation not only influences our emotional state, but also shapes our actions and choices. Indeed, certain challenges pertaining to mental well-being arise from the struggle to adapt, exemplified by the difficulty in altering one's perception of a situation due to persistent negative thoughts. In an effort to tackle these concerns, a groundbreaking study led by Dartmouth has successfully distinguished between the brain's activity in generating emotions and its activity in regulating emotions. This study, one of the pioneers in this field, has been published in the esteemed journal Nature Neuroscience. Lead author K.E. Bowe, a postdoctoral researcher in the Cognitive and Affective Neuroscience Lab, CAN-Lab, at Dartmouth, expresses enthusiasm as a former biomedical engineer in discovering brain regions exclusively dedicated to the regulation of emotions. This groundbreaking finding sheds light on the mechanisms of emotion regulation and offers potential clinical applications through the identification of specific targets. One possible application of the systems identified by the researchers is their potential as effective targets for brain stimulation, which could be utilized to improve the regulation of emotions. The University of Pittsburgh's Peter Giannaros provided the researchers with two separate sets of fMRI studies, which were analyzed using computational techniques. During the studies, participants were exposed to images that were expected to elicit negative responses, such as gruesome scenes or intimidating animals, while their brain activity was monitored in an fMRI scanner. Following the presentation of a stimulus, the participants were instructed to generate alternative thoughts about an image to reframe it and reduce its aversiveness. This was followed by the introduction of a neutral image and finally another image that was disliked. Through the analysis of neural activity, scientists were able to distinguish the specific regions of the brain that exhibit higher levels of activity during the regulation of emotions compared to the generation of emotions. Through the recent study, it has been uncovered that the process of emotion regulation, scientifically referred to as reappraisal, engages specific regions within the anterior prefrontal cortex and other cortical hierarchies associated with higher-level cognitive processes. The precise identification of these areas in relation to emotion regulation is a novel finding. These regions, which play a crucial role in abstract thinking and the formation of future-oriented mental representations, are also involved in various other cognitive functions at a higher level. The greater the capacity individuals possess to engage these specific regions of the brain responsible for regulating emotions, the stronger their ability to withstand negative experiences without allowing them to have a personal impact. These discoveries further support previous studies that have connected these brain areas to improved mental well-being, as well as the capacity to resist temptations and steer clear of substance abuse. The findings additionally showcased that the amygdala, recognized as the region of the brain associated with threat and negative emotions, and traditionally viewed as an archaic subcortical center for danger, reacts to unpleasant encounters in a consistent manner, regardless of whether individuals are employing cognitive strategies to lessen negative emotions or not. The cortex is truly accountable for producing emotional reactions in individuals, as it alters our perception and interpretation of events in our surroundings, explains Bo. In addition to their primary focus, the researchers were intrigued by the identification of the neurochemicals that engage with systems responsible for regulating emotions. Neurotransmitters such as dopamine and serotonin play a vital role in modulating communication between networks of neurons and are targeted by both illicit substances and therapeutic interventions. Certain neurotransmitters may play a crucial role in facilitating the capacity for self-regulation or the process of down-regulating. By examining the brain maps associated with emotion regulation in both datasets, the team conducted a comparison with neurotransmitter binding maps from 36 additional studies. It was found that the systems responsible for regulating negative emotions shared common ground with specific neurotransmitter systems. According to Tor Wager, the Diana L. Taylor Distinguished Professor in Neuroscience and Director of the Dartmouth Brain Imaging Center at Dartmouth, our findings indicate that regions associated with emotion regulation contain a significant concentration of receptors for cannabinoids, opioids, and serotonin, including 5-HT2A. This observation raises concerns about the potential long-term impact on our ability to self-regulate when drugs that interact with these receptors are consumed. It suggests that the drugs primarily influence the emotion regulation system, prompting further inquiry into their lasting effects. The role of serotonin in depression is widely recognized, as it is targeted by the majority of commonly prescribed antidepressant medications to prevent its reuptake in synapses, which are responsible for transmitting signals between neurons. The impact of psychedelic drugs on mental health is primarily centered around the serotonin receptor known as 5-HT2A. A recent study has revealed that these drugs alter our perception of life events and our capacity for self-regulation, which may contribute to their effectiveness in treating depression and other mental disorders. It is crucial to note that without proper psychological support, particularly in the case of psychedelics, these drugs are unlikely to yield positive results. By shedding light on the necessity of combining psychological and pharmaceutical interventions, this study has the potential to enhance therapeutic strategies and promote integrated treatments. According to Wager, it is crucial to take into account these particular associations that arise from fundamental scientific research. Gaining insight into the effects of drugs necessitates comprehending the neural mechanisms at play and their impact on cognitive functioning. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.